Fine, let's start. Number 1. The beetle inside the dung. Imagine naming a brand new species of animal, an entire creature unknown to science, and discovering it not in a slab of rock, not in a block of amber, but inside a piece of fossilized excrement. That is exactly what happened with a tiny beetle called Triomyxa coprolithica. Its species name is not subtle. Coprolithica means, quite literally, of the coprolite. A coprolite is fossilized feces, a turd turned to stone, and the word itself was coined by the English geologist William Buckland back in 1829 from the Greek words for dung and stone. The beetle is about 230 million years old. That places it in the late Triassic, a time long before the famous giants of the Jurassic and Cretaceous, when the first dinosaurs and their close relatives were only beginning to spread across the planet. The dung came from a site called Krasiejów in the Silesia region of Poland, a celebrated Triassic bone bed packed with the remains of early reptiles and amphibians. So, who produced the dropping? The leading suspect is an animal called Silosaurus apolensis. It was a slender dinosaur form, a dinosaur precursor, about 2.3 meters long, with a beak at the tip of its jaws. That beak is a clue, because it suggests an animal that pecked and picked at small food items. And inside its fossilized dung, researchers found beetles. The discovery was published in the journal Current Biology in 2021 by Martin Kvarnström, Martin Fikacek, Per Ahlberg, and their colleagues. To see what was hidden inside the stony dung, they used a technique called synchrotron microtomography at the European Synchrotron Radiation Facility in Grenoble, France. A synchrotron is a vast machine that accelerates particles to produce extremely intense beams of light, and those beams can scan the interior of a solid object at the scale of a few millionths of a meter. The result was a set of three-dimensional reconstructions of the beetles trapped inside, complete with legs, antennae, and individual body segments. This is the part that stuns specialists. The beetles were not flattened smears. They were preserved in 3 dimensions, almost whole. At least one nearly complete beetle was recovered, along with fragments of dozens of individuals, all from a single dropping. Each beetle was about 1 millimeter long, the size of a speck, matching modern beetles of the suborder Myxophaga, which live today in wet films of algae. Triomyxa was placed in its own newly created family, an entirely extinct branch of the beetle tree. Now consider what this means for our usual picture of exceptional fossils. For small, delicate creatures, the gold standard of preservation has always been amber, fossilized tree resin that traps insects in clear golden tombs. But amber only formed where resin-producing trees grew, and most amber with insects inside is from the Cretaceous or younger. Dung, by contrast, was produced everywhere, by everything, all the time. Kvarnström and his team argued that coprolites might rival or even exceed amber as an archive of tiny ancient life, and this Triassic example pushes that kind of pristine, three-dimensional insect preservation tens of millions of years deeper into the past. How did a soft little beetle survive at all? The answer is chemistry. The dung mineralized rapidly, with calcium phosphate replacing and binding the organic material before it could rot away. That phosphate effectively cast the beetles in stone, locking their hard outer skeletons in place. And the way the beetles were swallowed, whole and in bulk, tells us something about Silosaurus itself. This was an animal that gulped small invertebrates rather than chewing them finely, which means it was at least partly an insect eater, complicating older ideas that it was a strict plant eater. It is worth dwelling on how the beetles came to be there in the first place. Silosaurus seems to have hunted in wet, marginal environments, the kind of damp algal mats where tiny Myxophaga still thrive today, snapping up mouthfuls of substrate dense with insects. Most of those beetles were chewed or broken on the way down, which is why the dropping holds fragments of dozens of individuals. But a few were swallowed so cleanly that they survived intact, sealed and scanned 230 million years later. That gives us not just a beetle, but a behavior. The feeding habits of a dinosaur precursor reconstructed from the bodies of its prey. The most remarkable detail of all may be the most human one. These coprolites had been sitting in collections for years before anyone thought to scan their insides. The beetle was always there. We simply had not looked. It is a reminder that some of the most spectacular fossils in the world are not waiting to be dug up, but waiting to be re-examined, hidden inside specimens we already hold. Number 2. The grass that should not exist. For a long time, the story of grass and the story of grazing animals were told as a single tale. Grasses spread, open plains appeared, and animals with the right teeth evolved to eat them. Somewhere around 55 million years ago, well after the dinosaurs were gone. Then someone looked inside dinosaur dung and the timeline fell apart. The discovery was reported in the journal Science in 2005 by Vandana Prasad, Caroline Stromberg, Habib Ali Mohammadian, and Ashok Sani. They studied coprolites from the Lameda beds of central India, sediments laid down in the latest Cretaceous, roughly 67 to 65 million years ago. These droppings are attributed to tyrannosaurs, the enormous long-necked sauropods that were the dominant large plant eaters of India at the time. Inside the dung, the researchers found phytoliths. A phytolith is a microscopic body of silica, essentially natural glass, that many plants build inside their own tissues. Different plant groups make differently shaped phytoliths, so these tiny glass particles act like fingerprints for the plants an animal ate. And some of the phytoliths in the tyrannosaur dung were unmistakably the type made by grasses, the plant family known as Poaceae. That single observation rewrote a chapter of botany. Before this study, the oldest solid evidence for grasses sat at about 55 million years ago. The Indian coprolites pushed the origin of grasses back by at least 10 to 12 million years, into the age of dinosaurs. Worse for the old story, the phytoliths represented several distinct kinds of grass, which means grasses had not just appeared, but had already begun to diversify before the asteroid struck. The expert credentials here matter, because the claim was bold. Caroline Stromberg is one of the world's leading specialists in phytoliths and the evolution of grasses, and her identification gave the result real authority. Even so, critics initially worried the grass particles might be modern contamination. The defense was physical. The phytoliths were embedded within the coprolite matrix itself, sealed inside the fossil dung, not dusted on the surface. And the timing fit independent molecular clock estimates, calculations based on the rate of genetic change, which had already hinted that grasses might be older than their fossil record suggested. Here we can lay out the competing interpretations side by side. The mainstream reading is straightforward. Grasses evolved earlier than the body fossil record showed, and some dinosaurs ate them. A more cautious traditional view held that the phytoliths could be intrusive or misidentified, and that grasslands as ecosystems still came much later. And a more speculative angle imagines herds of dinosaurs grazing on early grasses, much like later mammals, an image that goes beyond what the dung alone can prove. The evidence supports the first view most strongly, while reminding us that a few microscopic particles cannot, by themselves, conjure a prehistoric prairie. What makes this discovery so durable is the toughness of the evidence. Phytoliths are made of opaline silica, and silica survives both digestion and fossilization with ease. Soft plant tissue almost never fossilizes, but the glass skeletons inside that tissue can endure for tens of millions of years, passing through a dinosaur's gut and into the rock. The same coprolites also held phytoliths from palms, conifers, and other plants, painting a picture of a broad, mixed diet. Tyrannosaurs did not have grinding, grazing teeth, so they likely cropped the grass and swallowed it whole, relying on a vast fermentation chamber of a gut to extract the nutrients. There is a deeper consequence here, one that reaches into the evolution of mammals. For decades, the spread of grasslands was tied to the rise of grazing animals like horses, with their high-crowned, ever-growing teeth built to resist the abrasive silica in grass. If grasses were already present and diverse in the Cretaceous, then the plants and their silica defenses had a long head start, evolving alongside dinosaurs long before any mammal specialized to eat them. The droppings, in the end, preserved what no skeleton ever could, a record of the plants that fed the largest animals to walk the land, and a clue to an evolutionary arms race that would not fully play out for tens of millions of years. Number 3. The vegetarians that ate shellfish. We like our categories clean. Plant eaters eat plants. Meat eaters eat meat. But the contents of certain fossil droppings suggest some dinosaurs did not read the rulebook. In 2017, in the journal Proceedings of the Royal Society B, Karen Chin, Rodney Feldman, and Jessica Tashman described coprolites from the Kaiparowitz Formation of southern Utah, rocks about 75 million years old. The dung came from large plant-eating dinosaurs, most plausibly hadrosaurs, the duck-billed dinosaurs that were abundant in those beds. Inside the plant-rich dung were the broken shells of crustaceans. Researchers identified them by the distinctive layered microstructure of the exoskeleton, the prismatic and laminated calcite that decapod crustaceans build into their shells. These were likely crabs or crab-like animals, and the fragments were several millimeters across, far too large to have been swallowed by accident, along with a mouthful of leaves. Karen Chin of the University of Colorado Boulder is the leading authority on fossil dung, and her interpretation was careful. The dinosaurs, she argued, were eating these crustaceans on purpose, probably as a seasonal source of protein and calcium tied to the demands of breeding and egg-laying. This is not a wild idea. Many modern plant-eating birds deliberately consume insects, snails, and grit during their reproductive season because building eggs requires far more calcium than a leafy diet provides. The crustaceans did not arrive alone. The same coprolites contained rotting conifer wood and traces of fungus, which points to a single feeding strategy. The dinosaurs were targeting decaying logs, and those logs were home to the crustaceans. Rodney Feldman, a specialist in fossil crustaceans, confirmed the identification of the shell fragments, and crucially, the crustaceans turned up in more than one coprolite. This was a repeated pattern, not a one-time accident. The implications ripple outward. The strict line between herbivore and carnivore begins to blur into something biologists call facultative omnivory, the ability to eat across categories when the body demands it. The duck-billed dinosaurs look less like simple grazing machines and more like flexible generalists, exploiting whatever the forest floor offered. And it suggests they had the nutritional adaptability we see in modern large herbivores under physiological stress. The coprolites themselves were large, up to tens of centimeters, exactly what you would expect from animals weighing several tons. Once again, the dung recorded a behavior that teeth and bones alone would never have revealed. The strange truth that some of the great plant eaters of the Cretaceous occasionally cracked open a crab. Number 4. A taste for decay. If eating shellfish was surprising, the staple diet hidden in these same hadrosaur droppings is stranger still. In work published in the journal Paleontology in 2007, and in related studies, Karen Chin showed that coprolites from the Two Medicine Formation of Montana, around 75 million years old, were dominated not by soft leaves, but by conifer wood. And it was not fresh wood. It was rotten. The wood fragments showed clear signs of fungal decay, riddled with the borings of small organisms and the traces of fungal threads called hyphae. In other words, the duck-billed dinosaurs were deliberately eating decomposing logs. Chin proposed that they did this because decay actually made the wood more useful. Fungi break down the toughest components of wood, the lignin and cellulose, partially digesting it before the dinosaur ever took a bite. Rot also concentrates nutrients and packs the wood with protein-rich fungi and invertebrates. The dung is attributed to large hadrosaurs such as Maiasaura, which are well known from the same Montana beds. Some of these coprolites were enormous, with an inferred original volume of more than 7 liters, the unmistakable output of a multi-ton animal. And the find was genuinely unexpected, because hadrosaur teeth, with their complex grinding batteries, had always suggested a diet of soft foliage, not hard, woody material. Why would an animal built for leaves turn to rotting logs? The most likely answer is season. When preferred soft vegetation became scarce, perhaps in a dry season, the dinosaurs may have switched to the reliable, if unappetizing, resource of decaying wood. To digest such material, they would have needed a gut full of fermenting microbes, much like modern animals that process tough plant matter in a large hindgut. The wood fragments in the coprolites are sized and oriented in ways consistent with passage through exactly that kind of large fermentation system. This discovery did more than add an item to a dinosaur menu. It established coprolites as a tool for reconstructing entire trophic interactions, the web of who eats what. The rotting wood connected the dinosaurs to the fungi that decayed it, to the invertebrates that lived in it, and, as we will see, to the insects that would later recycle the dung itself. Chin examined all of this using petrographic thin sections, slicing the coprolites as thinly as one would slice a rock, and studying them under a microscope. The cellular structure of the wood was preserved well enough to identify it as conifer. The picture that emerged was of dinosaurs deeply embedded in the forest's cycle of decay, not standing apart from it, but feeding directly on its rot. Number 5. The parasites within. So far, the dung has told us what dinosaurs ate. It can also tell us what was eating them from the inside. In a study published in the journal Parasitology in 2006, George Poinar Jr. and Arthur Bucco reported the remains of parasites preserved in coprolites, attributed to early Cretaceous reptiles, with material associated with sites including Belgium. Inside the fossil dung were eggs and cysts. The researchers identified structures interpreted as belonging to trematodes, which are flukes, to nematodes, which are roundworms, and to single-celled protozoans. Some of the specimens preserved oocyst-like structures that resembled those produced by coccidian protozoan parasites, a group that still infects reptiles and birds today. The famous Belgian locality of Bernissart, which yielded a herd of complete iguanodon skeletons, is linked to some of this material. George Poinar Jr. of Oregon State University is one of the world's foremost experts on fossil parasites, best known for his work on creatures trapped in amber. The eggs he and Bucco studied were identified by their characteristic shapes, sizes, and the ornamentation of their shells, examined under high magnification. Some nematode eggs measured only a few tens of micrometers, smaller than a dust mote, demanding careful microscopy to recognize. The significance is direct and a little unsettling. This is fossil evidence that dinosaurs and their reptilian contemporaries carried internal parasites, suffering the same kinds of gut infections that plague modern animals. The protozoan cysts hint at diseases comparable to coccidiosis, a real and sometimes deadly affliction of today's birds and reptiles. And the find connects to other clues from the Cretaceous, including biting insects preserved in amber that may have transmitted blood parasites, building a picture of a Mesozoic world teeming with disease. Preservation, again, came down to speed. The dung had to mineralize rapidly to seal the delicate parasite eggs before they decayed. The eggs survived in part because their shells are made of unusually durable proteins, far tougher than the soft bodies of the worms that produce them. That is a recurring lesson of paleoparasitology, the study of ancient parasites. We rarely find the parasites themselves, but we find their armored eggs, and those eggs reveal more than disease. Because many parasites cycle through multiple hosts, their presence can hint at a dinosaur's diet and its place in a web of predators and prey. Consider what a parasitic life cycle actually requires. A fluke or a roundworm often passes through an intermediate host, a snail, an insect, a fish, before reaching its final host. So the mere presence of a particular parasite egg can imply that the dinosaur was eating, or being bitten by, the creatures that carried it. A single egg becomes a thread that, pulled gently, unravels part of an entire ancient ecosystem. The dung reframes these giants not as isolated monsters, but as walking, breathing ecosystems, each one a habitat for creatures we can still find sealed in stone. The animals we picture as invincible were, like every living thing, hosts to a hidden world of smaller life, feeding on them from within. Number 6. The ecosystem inside the feces. A fresh pile of dung, in any age of the earth, is not waste. It is an opportunity, and the creatures that exploit it leave their mark. In a study published in the journal Paleos in 1996, Karen Chin and Bruce Gill described something extraordinary inside large herbivore coprolites from the Two Medicine Formation of Montana. They found burrows, tunnels driven through the dung, and then backfilled with sediment and pellets of dung. These are interpreted as the work of dung beetles, members of the family Scarabaeidae. The implications of those tunnels are vivid. For beetles to burrow through the dung, it had to be fresh and soft, which means the dung was discovered and colonized within hours to days of being deposited, before it dried and hardened. The beetles tunneled into it, fed on it, and very likely nested in it, just as their descendants do today. This is among the oldest direct evidence of dung beetle behavior anywhere in the fossil record, pushing the existence of this entire ecological guild back into the Cretaceous. Bruce Gill, a specialist in beetles, helped confirm that the dimensions and backfilled structure of the burrows matched the feeding traces of modern tunneling dung beetles. The coprolites themselves were large, the product of multi-ton hadrosaurs, which would have generated a steady supply of dung for the insects to exploit. It is worth pausing on what this tells us. Modern dung beetles are essential recyclers, returning nutrients to the soil and clearing the landscape of waste. This discovery shows that the same crucial role was being performed alongside the dinosaurs, more than 70 million years ago. It implies a kind of coevolution between enormous herbivores producing prodigious amounts of dung and the small industrious insects that depended on it. There is a beautiful subtlety here. No beetle bodies were preserved in these coprolites at all. What survived was only their architecture, the tunnels they dug, a category of evidence that scientists call trace fossils, or ichnofossils. From empty tunnels alone, researchers reconstructed an entire behavior. The dung came from the same formation that preserves Maiasaura nesting grounds, tying the droppings to known producers and to the broader life cycle of the herd. And it raises a haunting question for the end of the Cretaceous. Dung beetles today rely heavily on mammal dung. When the great dinosaurs vanished, what happened to the insects that had built their lives around their waste? The answer, as far as we can tell, is that the dung beetles adapted, shifting from the colossal droppings of dinosaurs to the smaller, more frequent dung of the mammals that inherited the world. The guild survived a mass extinction by changing its menu. There is a striking continuity in that, a chain of nutrient recycling unbroken from the age of Maiasaura to the cattle pasture of today. The same backfilled tunnel that a beetle dug into a hadrosaur's dropping 75 million years ago has a direct descendant in the burrows beneath a modern cowpat. The fossil dropping, it turns out, preserves not just a meal, but an entire miniature world, and the deep history of one of the planet's most important and least celebrated ecological jobs. Number 7. The meal caught mid-digestion. Some fossil droppings record diet with almost gruesome clarity because the prey passes through barely changed. Across many formations, the coprolites of carnivores and fish eaters preserve undigested fish scales, fin spines, and fragments of bone, a direct snapshot of the last meal. This evidence sits at the center of one of the great dinosaur debates, the diet of the spinosaurids. Spinosaurus, Baryonyx, and Suchomimus are long-snouted predators, long suspected of eating fish, and the contents of dung and gut cavities support that idea. The holotype skeleton of Baryonyx walkeri, found in the Wealden Rocks of Surrey in England and studied by Alan Charig and Angela Milner, preserved the scales of a fish called lepidotes in the region of its stomach, complementing the wider coprolite evidence. Fish scales are surprisingly easy to recognize in fossil dung. Ganoid fish like lepidotes had thick, glossy scales coated in a hard enamel-like substance called ganoine, often in a distinctive diamond shape. That enamel-rich coating resists the acids of a digestive tract far better than soft tissue does, which is exactly why the scales survive the journey through a predator's gut and into a coprolite. Many of these scales show acid etching on their surfaces, a chemical signature of having passed through a stomach. The droppings of crocodile relatives and other aquatic predators turn up across many sites, carrying fish bone and scales, and the Wealden and Bernissar deposits yield reptile coprolites with similar contents. Some predator coprolites even preserve a spiral or scroll shape, the imprint of a spiral valve intestine, an anatomical feature found in sharks, some fish, and certain reptiles. That shape is a clue not just to diet, but to the internal anatomy of the animal that produced it. We can again weigh competing interpretations. The mainstream view holds that spinosaurids were specialized fish eaters, supported by fish-bearing coprolites, by the shape of their crocodile-like snouts, and by chemical isotope studies of their bones. A more cautious view points out that finding fish in a few droppings does not prove an animal ate only fish, and that these predators were probably opportunists, taking whatever they could catch. A bolder, more speculative reconstruction casts Spinosaurus as a fully aquatic, swimming hunter, an image still hotly contested among paleontologists. The dung supports fish eating clearly, but how much and how aquatic remains open. What is certain is that few fossils are as honest as a coprolite full of someone else's scales. Number 8. Soft tissue that survived the gut. The contents of a dropping tell us what an animal ate. The condition of those contents tells us how its body worked. When Karen Chin analyzed large carnivore coprolites, she found not only crushed bone, but traces interpreted as muscle and connective tissue, soft material that had somehow survived passage through a giant predator's digestive system. This is genuinely strange, because soft tissue is the first thing to dissolve. Its survival implies that food moved through the gut relatively quickly, transiting before the stomach acids could fully break it down. Under the scanning electron microscope, bone fragments in the dung still retained partly preserved organic and even cellular structure, and fibrous microstructures distinct from the surrounding matrix were interpreted as remnants of muscle. The implications reach into tyrannosaur physiology. If a tyrannosaur fully macerated its food in acid for long periods, the way some scavengers do, we would expect soft tissue to vanish and bone to be heavily etched. Instead, the bone showed minimal etching and flesh made it through. That points to a relatively fast gut transit, a digestive style quite different from that of mammalian carnivores. The mixture of crushed bone and muscle also suggests the animal was bolting down chunks of flesh and bone together, rather than carefully stripping meat from a carcass. There is a useful modern comparison in the hyena, which also swallows and passes large amounts of bone. But the dinosaur evidence suggests an even faster throughput, and the very preservation of soft tissue in feces is exceptional, requiring extraordinarily rapid mineralization, helped along by the calcium phosphate released from all that digested bone. The chemistry that hardened the dung was, in a sense, supplied by the meal itself. This kind of finding feeds directly into a long-running argument about Tyrannosaurus rex, whether it was primarily an active predator or a scavenger, because the way an animal processes food relates to how it gets that food. it also sits within a broader and sometimes controversial body of research on organic remains preserved in Mesozoic fossils. What is not in doubt is the value of the specimen. From a single petrified dropping, scientists extracted information not just about a tyrannosaur's last meal, but about the inner workings of its gut, a rare and intimate glimpse of physiology from an animal dead for tens of millions of years. Number 9. The coprolite that proved a king ate bone. Of all the fossil droppings ever studied, one stands above the rest, and it belongs, almost certainly, to Tyrannosaurus rex. It was described in the journal Nature in 1998 by Karen Chin, Timothy Tokarik, Gregory Erickson, and Lewis Kalk, and it remains the most famous coprolite in the world. The specimen came from the Frenchman Formation of southwestern Saskatchewan in Canada, deposited in the latest Cretaceous around 66 million years ago, right at the edge of the dinosaur's final chapter. It is about 44 centimeters long, with an estimated original volume of more than 2 liters. At the time of its discovery, it was by far the largest carnivore coprolite known, and the only carnivore in that formation large enough to have produced it was Tyrannosaurus rex. The contents were the revelation. Between 30 and 50% of the coprolite's volume is fragmented bone. This was direct physical proof that Tyrannosaurus did not simply tear off and swallow flesh. It crushed bone, shattered it, and swallowed the pieces, a feeding behavior called osteophagy. The bone fragments are angular and show only light acid etching, which, as with the soft tissue evidence, points to relatively rapid passage through the gut. The bone is thought to have come from a young plant-eating dinosaur, perhaps a juvenile of a horned or duck-billed species. This settled a real scientific debate. Researchers had argued for years about whether any large theropod actually consumed bone, the way modern hyenas do, or whether they delicately stripped meat and left skeletons behind. The Saskatchewan coprolite ended the argument. Gregory Erickson, a specialist in the mechanics of dinosaur bites, had separately shown that Tyrannosaurus possessed a bite force capable of shattering bone, and here was the digestive proof that it used it. The sheer amount of surviving bone showed the animal could not fully dissolve it before excreting it. Timothy Tokarik of the Royal Saskatchewan Museum was central to recovering and caring for the specimen, and the bone content was confirmed through both petrographic thin sections and scanning electron microscopy, ruling out the possibility that the bone was simply contamination from the surrounding rock. The coprolite lacked a strong spiral shape, consistent with the non-spiral gut of a theropod dinosaur. A second, even larger Tyrannosaur coprolite was later reported, reinforcing the pattern. More than any other single specimen, this petrified dropping rebuilt our image of Tyrannosaurus rex as a destructive, bone-crunching feeder, and it proved, once and for all, that fossil dung can answer questions that bones alone never could. Number 10. The record breaker. We end on sheer scale, with a specimen nicknamed Barnum, the largest coprolite of its kind ever formally recognized. It is tied to the collector and researcher George Franson, who holds a Guinness World Record for the largest collection of coprolites on Earth, numbering well over 1,000 specimens. His record-setting dropping measures more than 67 centimeters long, about 26.5 inches, and was recognized by Guinness World Records in 2020 as the largest coprolite from a carnivore. Here, honesty demands a caveat, and it is an important one for understanding the whole field. Barnum is not a dinosaur dropping. It dates to the Paleocene, the epoch after the dinosaur's extinction, and it is most likely the work of a very large crocodilian. The nickname itself nods both to the showman P.T. Barnum and to the legendary fossil hunter Barnum Brown. That a crocodile relative could leave a dropping larger than any confirmed Tyrannosaurus coprolite is a lesson in itself. The biggest fossil feces are not always dinosaurian, and telling them apart is one of the hardest problems in the science. This difficulty is real. large crocodiliform droppings can closely resemble dinosaur dung, and where these giant coprolites preserve a spiral or scroll structure, that shape actually points toward a spiral valve gut, the kind found in crocodilians and some fish, rather than in most dinosaurs. To assign a producer responsibly, researchers must match the specimen's size, its internal anatomy, and the geographic range and time period of the candidate animals. Barnum checks the boxes for a large Paleocene crocodilian, not a dinosaur. What makes giant coprolites scientifically valuable beyond their shock value is their volume. The bigger the dropping, the more it can preserve. More bone, more scales, more microscopic clues about diet and digestion. Franson has turned his collection into a tool for public engagement, founding a museum dedicated entirely to coprolites, playfully called the Pooseum, where specimens like Barnum teach visitors about digestion, diet, and the strange process of fossilization. The Guinness recognition did real work, bringing mainstream attention and a measure of legitimacy to a subject many people assume is a joke. Barnum closes our countdown not as a dinosaur, but as a reminder of just how large and how informative a humble fossil dropping can be. To understand why any of these discoveries exist, we have to ask a stranger question. How does something as soft and perishable as dung survive for tens of millions of years? Coprolites are trace fossils, recording an animal's behavior rather than its body, and their survival depends on a rare and demanding sequence of events. The first requirement is speed. Dung must be buried quickly, before scavengers, insects, weather, and microbes can tear it apart. Most droppings, of course, vanish within days. Only a vanishing few find the right conditions. The crucial chemistry is mineralization by calcium phosphate, which permeates the dung and binds its contents into solid rock. This is why carnivore droppings fossilize so much more readily than those of plant eaters. A meat eater's dung is rich in digested bone, and that bone supplies the very calcium phosphate that hardens the dropping from within. It is no accident that so many of the most famous coprolites belong to bone eaters and fish eaters. The diet that produced them also preserved them. Herbivore dung, lacking that mineral wealth, is far more fragile, which makes Karen Chin's wood-rich and crustacean-bearing specimens all the more precious. Quiet, oxygen-poor, low-energy environments help, slowing decay long enough for mineralization to take hold. And the same phosphate that hardens the dropping is what casts its most delicate inclusions. The beetles, the parasite eggs, the fish scales, freezing them in 3 dimensions. Distinguishing a true coprolite from an ordinary mineral nodule can be tricky, and researchers rely on telltale inclusions, on characteristic shapes, and on the phosphate-rich chemistry. Spiral and scroll shapes betray a spiral valve gut, while pinched or coiled ends can preserve the very moment of excretion. Over deep time, the slow chemistry of diagenesis may alter a coprolite's color and mineral makeup, adding another layer the scientist must read through. This is also why the coprolite record is biased, and why we must read it with care. We see disproportionately the droppings of bone eaters and fish eaters from quiet, wet, low oxygen settings, because those are the conditions and diets most likely to fossilize. Vast amounts of dinosaur dung, especially the soft droppings of plant eaters in dry uplands, simply rotted away and left no trace. The fossils we hold are not a random sample of the past, but a filtered one, weighted towards certain animals, certain diets, and certain environments. A good coprolite scientist keeps that filter constantly in mind, never mistaking what happened to survive for the whole of what once existed. Every specimen in this video survived because of that rare convergence, rapid burial, the right chemistry, and stability across an unimaginable span of time. A fossil dropping gives up its secrets only to the right tools, and the transformation of coprolite science is really a story about technology. The oldest and simplest method is petrographic thin sectioning, slicing a coprolite as thinly as a sliver of rock and studying it under a microscope, the technique that let Chin identify rotten conifer wood inside hadrosaur dung. The scanning electron microscope goes further, imaging individual bone fragments, cellular structures, and the fine etching left by stomach acid. The most spectacular advance is synchrotron microtomography, the method that reconstructed the Triomyxa beetle in 3 dimensions without ever breaking the coprolite open. Other specialties answer other questions. Phytolith analysis reads the silica fingerprints of plants, as in the tyrannosaur grass discovery. Paleontology extracts pollen and spores to reconstruct ancient vegetation, and even the season of deposition. Biomarker and lipid chemistry can detect the molecular residues of diet, and energy dispersive X-ray analysis confirms the phosphate content that distinguishes a coprolite from a plain rock. What unites these methods is that each answers a different piece of the puzzle: diet, disease, season, or the identity of the producer. Just as importantly, the move from destructive techniques toward non-destructive scanning changed the culture of the field. Museums, once reluctant to let researchers saw apart rare specimens, now permit scans that leave the fossil intact and available for future study. Single droppings are now examined by teams that include entomologists, botanists, and parasitologists working side by side. The same families of instruments used to analyze Martian rocks and human tissue are now turned on fossil feces, and they have revealed that the most ordinary-looking lump can hide extraordinary detail. It was not luck that turned coprolites into a rigorous science. It was method. For all that these droppings reveal, they guard one secret jealously: the identity of the animal that produced them. This is the central, nagging problem of coprolite science. A tooth or a thigh bone carries the anatomy needed to diagnose its owner. A dropping carries no skeleton at all. Researchers are left to infer the producer from indirect clues: the size of the specimen, where and when it was deposited, the anatomy implied by its shape, and the contents sealed inside. Sometimes the inference is strong. The Saskatchewan coprolite was assigned to Tyrannosaurus rex because no other carnivore in that formation was remotely large enough to produce it. Other times, the honest answer is uncertainty. The giant specimen, Barnum, is most likely crocodilian, a vivid reminder that the largest droppings are not always a dinosaur's. We can frame the range of approaches as competing schools of thought. The mainstream cautious position attributes a coprolite only as far as the evidence allows, often settling for a description like large carnivore rather than a named species. A more historically minded approach leans on the associated bone beds and faunal lists of a site to narrow the field of suspects, and a more speculative, crowd-pleasing tendency reaches for confident one-to-one matches with famous species, a temptation that frequently overreaches the data. Several factors keep the question open. Spiral valve shapes can point toward fish or crocodilians and away from most dinosaurs. Many animals sharing one ecosystem can leave similar-looking dung. Contamination and the slow alteration of burial can muddy the contents. And some objects identified as coprolites have later turned out to be lifeless mineral concretions. This is why scientists often name coprolites with their own trace fossil classification, describing the form without committing to a maker. Far from being a weakness, this honest uncertainty is a strength. It keeps even the most compelling attributions appropriately tentative, and it leaves room for new finds to overturn old assumptions. The whodunit, in the end, is part of what makes the field so compelling. The science of fossil dung has a history as peculiar as its subject. It begins with the English clergyman and geologist William Buckland, who coined the word coprolite in 1829 and grasped, remarkably early, that fossil droppings could be read as records of ancient life. Buckland studied coprolites from the Jurassic and used them to reconstruct prehistoric food chains, an insight far ahead of its time. He was supplied with key specimens by Mary Anning, the brilliant fossil collector of Lyme Regis, who recognized that the stony objects she called bezoar stones were in fact fossilized feces. For much of the next century, Coprolites were treated as little more than curiosities, faintly scandalous oddities in museum drawers. There was, briefly, a more practical chapter. In 19th century England, coprolite-rich deposits were mined commercially for their phosphate, ground up to make fertilizer, and the so-called coprolite rush of the Cambridgeshire region fueled a small Victorian industry. But as scientific objects, droppings languished, regarded as too undignified to take seriously. That changed largely through the work of Karen Chin, whose studies from the 1990s onward transformed coprolites into a rigorous paleoecological tool. Her analysis of the Tyrannosaurus dropping and of hadrosaur dung showed the world that fossil feces could answer profound questions about behavior, digestion, and ancient ecosystems. George Poinar extended the reach of the science into disease, reading parasites from petrified dung. Vandana Prasad and Caroline Stromberg used coprolites to rewrite the evolutionary history of plants. And George Franson carried the subject to the public through his record-breaking collection and his cheerfully named Puseum. It is worth noticing the pattern in this history. At every stage, the obstacle to progress was not a lack of specimens, but a lack of seriousness, a sense that excrement was beneath the dignity of science. The breakthroughs came whenever someone was willing to set that squeamishness aside and ask what the dung could actually tell them. Buckland did it in a frock coat. Chin did it with an electron microscope. Each time, the reward was information available from no other source. The arc of this history runs from Anning's windswept cliffs to the beamlines of a synchrotron, from a Victorian punchline to a primary archive of vanished worlds. Each generation discovered new questions that dung, and only dung, could answer. Step back from the individual discoveries, and a larger truth emerges about why these unglamorous fossils matter so much. Coprolites preserve what almost nothing else can: behavior, diet, disease, and entire miniature ecosystems. The living detail that bones and teeth leave out. They are the only fossils that capture what an animal genuinely ate, frozen in the act of digestion. A skeleton tells us how a creature was built. A dropping tells us how it lived. The frontiers ahead are wide open. Researchers hope to reconstruct the full gut microbiomes of dinosaurs from chemistry locked in their dung. They want to measure how common omnivory really was among supposedly strict plant eaters after the surprises of crab shells and rotting wood. They are mapping the true diversity of Mesozoic parasites and diseases and asking how the great dung recycling food webs reorganized after the asteroid wiped out the giants that fed them. Each new scanning technique reopens old specimens for fresh discovery, and there may be more intact creatures, like the Triamixa beetle, waiting unexamined in museum drawers right now. The thread running through all 10 of these discoveries is that the contents we instinctively recoil from are precisely the most informative. Fossil dung connects predators to prey, plants to herbivores, insects to dinosaurs, and microbes to hosts, often within a single object held in the palm of a hand. It reframes the great extinct animals not as isolated monsters, but as living systems, embedded in webs of food, decay, and disease. The most overlooked fossil turns out to be one of the richest, a sealed report from a world we will never visit. The story of life, it seems, is written not only in bone and stone, but in what those vanished bodies left behind, ensuring that even the final, humble act of digestion can still speak to us across the depths of time.